Okay, the fourth and final type of macromolecule that's important in cells are the nucleic acids. And the reason that we talk about these is this basically is the genetic material of all living organisms, are these nucleic acids. So nucleic acids really, a lot of this class is going to be learning how we take the information, the material, the genetic material in a nucleic acid and turn it into a protein, and how those proteins really make your body work. Okay. So where do you find them? We know in our eukaryotic cells, and really all plants, fungi, protists, we find those nucleic acids in the nucleus of the cell, right? But your mitochondria actually have their own DNA. This is the DNA that's passed down. You get all of your mitochondria in the egg from your mother. The father does not contribute any mitochondria to your body, and so you can actually trace your maternal lineage through the DNA in the mitochondria. And then plants, similarly, they have chloroplasts that have DNA. In prokaryotic cells, these are bacteria. The DNA is found just in the cytoplasm. There's no nucleus that encloses it. Okay, types of nucleic acids. Okay. The biggest one is DNA. Okay. When we talk about the genome, that is all of the genetic content, all of the genetic information in your cells. Okay, so each cell has the same genome in it, but that's all of the information that really makes you who you are. Okay, which chromosomes we're going to see are inside of your nucleus. Your DNA is organized into these chromosomes, and the chromosome itself is made up of both DNA and these proteins that help the DNA to organize itself. So this, I guess, is just terminology. You should know genome means all your chromosomes. Chromatin is what a chromosome is made up of, which is DNA and proteins. And then a chromosome are the discrete individual threads of DNA. So we have 23 different threads, long strands. They pack up into this chromatin, right? And all of the information in all of those chromosomes is your genome. Okay. DNA is one nucleic acid, RNA is another. And we're going to talk about RNA later in the semester. Right? And we said all of this nucleic acid is there basically to be translated into proteins, and RNA is part of this process. So we have three basic types of RNA: the messenger RNA, or mRNA, tRNA, and rRNA. And trying to think what to say at this point is, I think just know those are three types of RNA. Know that mRNA means messenger, tRNA means transfer, rRNA means ribosome. Because we're going to spend a lot of time later talking about these individual macromolecules and their role in this important process of protein synthesis. Okay, DNA versus RNA. Well, both of them, they are polymers. They're big molecules, and each of those polymers is made up of monomers, just like we saw with all all of the macromolecules we're talking about here. Right? So here the monomer is the nucleotide. So the building block of the polymer are called a nucleotide. And each nucleotide is made up of these three things. Okay, nitrogenous base. Here are the bases at the top. The A, C's, T's, and G's, and U's, those are the bases. Those are attached to a pentyl sugar, 1 carbon, 2, 3, 4, 5 carbons, pentyl sugar. And then we have a phosphate group on the side. Okay. So these three things, a base, a sugar, and a phosphate, make up a nucleotide, which is the building block of a nucleic acid. Okay. Two main types of nitrogenous bases. There are the pyrimidines, right, which are the cytosines, diamines, and uracils, and then there are the purines, right, which are the guanine and the adenine. One of my previous students, he said he learned to discriminate or differentiate between these two is that Georgia is pure through and through, right? So Georgia GA is pure, right? So the G and the A make the purines, and then you can just put the other three into a group of the pyrimidines. Okay. Two types of sugars. We go back to this pentyl sugar. There is a deoxyribose and a ribose sugar. The difference not being really that important at this point, but you should know that there are two types, OSE, right? We talked about that convention about how that basically means it's a sugar. And of course, the D and DNA comes from this deoxyribose. The R and RNA comes from the ribose. These are the differences. One of the main differences between DNA and RNA is what type of sugar is found in which of the nucleic acids. Okay, DNA, double helix. The sides of this helix are the sugar and phosphate. The inside of the helix are the nitrogenous bases. So this is the sugar phosphate backbone, and then going across here, kind of like the rungs of the ladder, those are the bases. Yeah, I'm just introducing this molecule at this point. You will get to know the molecule very well at some point. Okay, a very good table to know. I don't have a place to put in here. Structure of the summary of the key features of DNA and RNA. So you'll want to know this table. You'll want to know the difference between them. Okay, DNA carries the information. RNA helps us to use that information to make the protein. DNA is found in the nucleus. RNA actually goes from the nucleus out to the ribosome, where the proteins are actually made. Right, so we said mRNA was the messenger RNA. That's the messenger that leaves the nucleus and goes to the ribosome. DNA, double helix, RNA, single strand. DNA is made up of deoxyribose. RNA is made of ribose sugar. DNA, really, the pyrimidines, the thymine is replaced by uracil in RNA in the pyrimidines. In the purines, they're the same in both DNA and RNA. They're the G and the A. Okay, so just know those differences. Okay, and that's it. Okay, so wish there was a big overall table that compared the 4 types of macromolecules, right? You should know the monomers for each of them. You should know the type of covalent bond that is formed between the monomers in each of them. You should know the function of each of those macromolecules, and you should be able to recognize the actual chemical structure